Hi. Today I'll be talking to you about autism treatment, the cell danger response, and the SAT1 clinical trial. Here are my disclosures that you can look over on the Synchrony website. And here are some references and citations for background reading if you're interested in looking at more information about Suramin or the cell danger response, anti-purinergic therapy, or the metabolic features and regulation of the healing cycle. So the big question that I'd like to ask is, are the behavioral symptoms of autism caused by a treatable metabolic syndrome? Now, for nearly 100 years, we've been cataloging the various triggers and causes of autism spectrum disorder. And these can be thought of as different mallets that can be used to strike the same cellular bell. And so ultimately, early life stress events, genes, air pollution, flame retardants, metals and pesticides, infections, fever, various cellular dysfunction states and mast cells and T cells and NK cells and cytokines in the microbiome all can contribute to ASD, but not in the same child. Okay, so sometimes they're more influential effects by these in individual children. But ultimately, they strike the same cellular bell that we have called the cell danger response that can lead to autism and myalgic encephalomyelitis and chronic fatigue syndrome and many, many other disorders. So over the last 100 years, we have really focused on the diversity of triggers, when I would say that the new chapter that we're opening up is really revealing the shared cellular response. So Grunya Sukareva was the first person to actually publish a description of autism spectrum disorder of 6 males in 1926 in a Russian publication. And then the more well-known child psychiatrist, Leo Kanner, published his first 11 cases in 1943. But ultimately, what I'd like to emphasize in this talk is really what we're looking at is the unity of the cellular response. So over the past 100 years, the metabolic features of autism and the cell danger response have been found to be the cell danger response, and the same biochemical pathways are regulated by biological, chemical, physical, and genetic stress. So one way to think about this, if you look at all the different kinds of responses in terms of neurotransmitters like tryptophan, serotonin, GABA, oxytocin, and then some of the exogenous environmental chemicals like Teflons and food preservatives, phthalates, dioxins, parabens, and then some of the endogenous responses that have to do with methylation changes, porphyrins, eicosanoids, oxalates, that change ultimately the structure of brain synapses and the cerebellum and the brainstem. Those are all connected by the features that we call the cell danger response. So in order to take the next step in understanding this, we ask the question, well, what is one of the most fundamental responses of the cell to environmental stress? And it turns out that extracellular ATP, or eATP, is a key regulator of the cell danger response. And so here's a cartoon of a cell with mitochondria in the nucleus, and then these channels in the membrane that are made up and part of purinergic receptor subunits like P2X7. This is a pennexin P2X7 channel that's regulated by oxidative changes as well as biophysical changes in the lipid composition of the membrane surrounding the pore. But anyway, when under stress, basically these channels open up in the membrane and cause ATP to leak. And this is a universal phenomenon. So all cells will release ATP in proportion to the amount of stress. And it's similar to the way you might think of a water balloon with thousands of little holes in the surface, releasing more water when the balloon comes under stress. and less water as the balloon as the balloon is under less stress. So mitochondria are constantly involved in making ATP, UTP, GTP in association with cytoplasmic metabolic pathways. But they communicate to the cell nucleus, and the nucleus, communicates back to mitochondria. And then when ATP is released and other metabolites that perform a signaling function that we call metabokines, will not only move out from the cell and then bind to receptors on the same cell of origin, but will also bind to receptors on neighboring cells. And so we went looking for ways that we might be able to pharmacologically reduce the amount of ATP loss. We call this the dissipative ATP loss. Because if ATP is lost from the cell to the outside, then there's less amount of ATP needed for growth and metabolism and normal cell functions, because that ATP, when it leaves the cell, becomes a signaling molecule that signals inflammation. So turns out there are drugs that can potentially block this, and we identified one that is called Suramin that I'll be able to talk to you about at the end of this. So the cell danger response is actually universal to all multicellular life on Earth. So all cells, all eukaryotic cells that have mitochondria, from plankton to people and plants, all have the same response. So here's a picture of a little mustard plant that has been engineered to fluoresce when its cells release calcium. Okay. And so it turns out that if you have a little injury over here on the right-hand side and you add either ATP or glutamate to that site of injury, then something very remarkable happens. The cell communicates that injury to the rest of the plant within a couple minutes. Okay, so it's a very rapid transfer. I'll let you see that one more time because it's an interesting response. But basically, injury gets signaled to all parts of the plant that are uninjured through this ancient signaling pathway involving ATP or glutamate. Okay, so ATP is found to be a modulating co-neurotransmitter at every different synapse. So here's a blood vessel, a capillary in the brain, and these are astrocytes, and this is a synapse that is releasing glutamate as the canonical neurotransmitter, but it turns out that every neurotransmitter also co-releases ATP in proportion to the amount of stress signaling that the body is undergoing at the time. Okay, so the more ATP released with the neurotransmitter, the greater the postsynaptic response. Okay. Just recently in this year, it's been found that even Fragile X has a root problem with ATP leak. And this is a citation from Elizabeth Jonas's group. But basically, here's a cartoon of the ATP synthase in mitochondria. And these subunits, the C subunits, can actually act as ATP channels that allow for release of ATP from mitochondria and then from into the cell and from the cell to the extracellular space that would then activate the CDR and inflammation. So one of the things I want to point out is ATP is made up of a sugar subunit called ribose, but also a nucleobase that's it actually includes adenine, but adenine is then can be deaminated to hypoxanthine and then oxidized to xanthine, which by itself, xanthine has been shown to be an anxiety-provoking signaling molecule that also can be involved in the production of hydrogen peroxide through an enzyme known as xanthine oxidase. So that sounds like an interesting phenomenon. Is there more known about that? So it turns out there is. And in, basically in the 20s and 30s, Karl von Frisch, who is a Nobel Prize-winning ethologist, characterized a material that he called Schreckstoff, which means the material that transmits a signaling of fear or alarm. And these turn out to be purine danger signals produced by a lot of different aquatic species, not just fish. But this is one example of hypoxanthine 3-inoxide that triggers an anti-predator behavior of innate inflammation and inflammation in a variety of different fish and other animals. And then xanthine has recently been shown to trigger a similar response in mice. It also influences T cell immunity and intensifies the aversive memories, the way that memory is laid down and retained in mammals. Okay, so I'm going to finish up by talking about the Suramin Autism Treatment 1 trial. This was a low-dose Suramin study, a single dose given by intravenous injection. Suramin can only be given by intravenous injection because if you give it by mouth, most of it just passes through and is not absorbed. So here's a little cartoon about the mechanism of the cell danger response and our approach to treatment. So over on the right is Bayer 205, which is actually the original name of Suramin. So here on the left are some mitochondria. And this is the following is going to be a description of a small phase 1/2 clinical trial at UCSD of Suramin, a 100-year-old drug that was developed originally to treat African sleeping sickness. But we wanted to test this hypothesis that autism might be a treatable symptom of cells. So here's a neuron, a nucleus, and mitochondria. And those mitochondria will make ATP that under stress conditions can be lost through opening up of these channels in the membrane. And when they do, they can bind receptors that send messages to the nucleus that trigger cell danger and the survival mode of those neurons. But when neurons are treated with Suramin, those channels get blocked and the danger signal of ATP is reduced. And so the cells can return more to a peacetime metabolism that allows healing and cell-cell communication in a more normal way. Okay, so this is a description of our design of the SAT1 trial. The children were paired by age, IQ, and autism severity, and then we prospectively randomized them into either the treatment group or a placebo group. It was completely double-blind. It was a study of 10 boys with ASD ages 5 to 14 years old and given a single dose of Suramin at 20 milligrams per kilogram IV or a saline placebo. And the outcomes were by the ADOS-2 score at 6 weeks after the infusion. So these are the 10 pioneers that were paired by age. So 11-year-old pair, 5, 14, 6, and 7. Now, 4 of these boys were nonverbal, had never spoken more than a single word at a time, never put 2 words together, and had severe oral motor dyspraxia. And as we began giving the infusion, something very remarkable started happening. We started noticing that half of the children started changing very significantly in many, many different ways, including language. But within days of his infusion, one boy looked up to his mother after dinner at the table and said, "I finished my dinner." Another boy who's had more language to begin with told his parents after coming home from school, "I learned to play tag with the kids at school today." This 14-year-old young man had not been able to speak in more than telegraphic single words, and his first he actually began to practice making many new sounds and actually singing and dancing, playing at home in new and different ways. Then after about a week, he went into the kitchen when his dad was making a sandwich for lunch, and his first words as a teenage boy were, I want to eat chips. This 6-year-old's first words were, I want to have a drink. And this young man's sentence that was unusual was to his mother as she was dropping him off for school. He said, today, I want to say hi to the crossing guard lady. So none of the other children started having any changes like this. And when we finally broke the code, we found that this group of 5 boys had received Suramin, this group had received placebo. When we looked at the numerical outcomes of this by ADOS and as by comparison score, raw social and restrictive repetitive movement scores, we found that all of these were very significantly improved in the Suramin group, but were not where there were no changes in the placebo group. And then when we looked at the aberrant behavior checklist and the autism treatment effect checklist, these were also significantly improved in the Suramin group, but not in the placebo. And so when we tallied up the ADOS scores and looked at the average score before and after treatment in the Suramin group, the average score was 8.6 initially, but the children who had received it 6 weeks later had an average score of 7.0. And so you can see the very significant difference between the error bars here. And the question for the next study, will 3 doses over 3 months help bring some of these children off spectrum? So many of you may know that the autism spectrum disorder is defined, in part by the ADOS score of 7 to 10. Okay, so anything below 7 is technically off the autism spectrum. Okay, and in the placebo group, you can see that there was no significant change. The error bars are completely overlapping, and they stay in this gray zone, which is the autism spectrum. Okay, so to summarize the results, so a single low dose of Suramin that achieved a 5 to 15 micromolar concentration in the blood was safe. And each of the 5 children who received Suramin showed improvements in all the core symptoms of autism. This was improved language, improved social interaction, and decreased stims and restricted interests. But in addition to that, each of the children also experienced non-core symptom improvements, and these included accelerated development and learning milestones at school, decreased anxiety and decreased meltdowns in novel situations, and improved sleep and interest in new foods, and improved GI function. And Suramin restored, we do know that from mice, although we didn't do this in the children, that in mice, Suramin restores the normal microbiome after a month of treatment. And there was actually a dramatic increase, a magnified benefit from the usual ABA, speech, and occupational therapies that the children were going to. And so none of the 5 children who received placebo instead of Suramin showed any of these improvements. So the next big questions: is Suramin safe and effective for the treatment of all kinds of autism in both boys and girls? We don't know this answer yet. And can Suramin help some children even come off the autism spectrum altogether? We don't know that either. And so the next study, which we will call the SAT2, is designed to test 3 doses over 3 months instead of just 1 dose, and see if we are able to continue the improvements that we saw in the first SAT1 study. So coming in 2021 is the SAT2 study. It's sponsored by a company called Kuzani Pharmaceuticals. That's a development company in the US to test Suramin for the treatment of autism. And we plan to test 60 to 80 children with autism at about 3 different sites. And Kuzani is just a development stage company in the US that is dedicated to making a new preparation of Suramin for the next autism clinical trials. But Suramin's also being developed for autism by another company called Pax Medica that's with ongoing trials in South Africa. And the newborn screening project is one that I really want to end with because we're very excited about the possibility of using a new test that we have developed in the laboratory to test dried blood spots that are collected from every child that's born in the United States by a heel stick at the time of birth. And those dried blood spots are then sent to sent for testing of a variety of for a variety of different genetic disorders. But we believe that we can combine both metabolic and exposome analysis. So exposome is the man-made environmental chemicals that are also present in these blood spots, and they're, we can measure 40 to 80 different environmental chemicals in all the blood spots that include Teflons and phthalates and flame retardants and a variety of other things. So what I would like to end with is a call for your help in this in conducting this new study, and we'd like to enroll 400 families, 200 with autism and 200 typically developing control families from who were born in California. Okay, because we're working right now with the California Universal Newborn Screening Program, but we want to expand this as soon as we can to all across America. But in order to do that, we need your help. So there are no new blood samples. All you need to do is go to our website, the naviaulab.ucsd.edu study, take a look, and if you qualify, you'll receive a $40 Amazon e-gift card as a token of our appreciation. It takes only 15 minutes of time. I stress again, there are no new blood samples. We are just testing the dried blood spots that were collected at the time of birth. And so we're looking for children 3 to 10 years of age. So I think that's the end of my talk today. And what would be the human impact? It would be priceless. So this is a child from our SAT1 study. And I wanted to thank all of the people who helped make this work possible, including the UCSD Christini Fund, the Brain Foundation, and CHARM, and many, many other groups from all around the world, over 2,000 people. So thank you very much. And I'll have time to answer some questions. Thank you.
Thank you so much. Super impressive results from that very small, granted, initial trial, but spectacular and inexplicable results. If there's not something, I mean, it really does indicate something really needs to be investigated there. The 2021 Kazani study, is that fully subscribed in terms of the people that are going to be?
No, the enrollment has not started yet.
Enrollment has not started. Is that the 200 people that you're looking for,
no,
or is that a different study?
Yeah, so the SAT2 study is the one that will look at 60 to 80 children with autism at 3 different centers in Southern California. Currently, that's probably still flexible at the moment, that's the one that was put off for a year because of COVID, and will start next year. The other study is just a dried blood spot study that has nothing to do with Suramin.
Okay.
So the one that I asked, people's help and participation on, click our website, take a look.
Yes.
You have a child 3 to 10 years of age, born in California.
That's the dried blood study.
That's the dried blood spot study, and that's completely separate.
Okay. And the Kazani effort, that's the study that is not yet begun enrollment and been put off.
Yeah, that's the SAT2. Kazani's the company, and they're the ones that are developing Suramin for the SAT2.
Okay, great. And is that's a California-based company, is it? Just out of curiosity?
No, they're incorporated out of Delaware, and they're a Boston company, but so they have a North American footprint.
I see a question here. Are there specific conditions like severe anxiety that you found in the subjects who had best improvements in expressive language? Or do you have any hypothesis about the mechanism of action in the language improvement case or cases?
Yeah, well, so the mechanism of action is just the one that we've always started out with, and that's basically this cellular basis of the cell danger response, and how that is mediated through a process that involves the release of ATP from stressed cells. And when that happens, ATP outside the cell acts as a cell danger molecule that can promote inflammation in the brain that takes the form of activated microglia. and it reduces the number of long distance contacts that occur during brain development in a child. And anybody who's interested in that, they should just, they could Google any of our work or PubMed any of our work on the cell danger response. That anti-purinergic therapy, it relates to this, this very ancient process by which all our cells determine whether the world is safe or threatening. And under conditions of threat, the priorities are completely reset. For example, when you're running away from a tiger, you're not going to be, thinking very hard about planning, let's say, the courses you want to take as a freshman in college. Okay?
The executive function disappears completely.
Right. So there's a new priority system, and so we view, a part of this, the questioner asked about anxiety. Anxiety we think of as absolutely fundamental, and it's a kind of cellular anxiety that is rippling up through many levels to produce
that actually leads to the next question, and actually it reminds me of the first diagram that you showed of all the possible insults or the things that strike that bell that cause the cell danger response. But the question is, what causes the cell danger response to chronically persist even when the original insult is removed? Any theories as to why it stays in that state?
Yeah, so that is a fundamental question that I've spent a lot of time on, and it comes down to breaking down the problem to two steps. So there's an initial what we call the initial threat or the initial injury that can be genetic, environmental, it can be ecogenetic, a combination of environmental and genetic factors that are specifically timed to vulnerable windows of child development in the case of autism. All those things can lead you to a condition that we call Autism Spectrum Disorder. But to be able to get back to a more normal trajectory for child development takes a completely different path. It's not sufficient to just remove the original insult. And we've actually studied this now in a dozen different diseases, ranging from chronic fatigue syndrome to post-treatment Lyme disease to even traumatic brain injury and major depressive disorder with and without suicidal ideation. Many different disorders are basically telling us the same thing, and that is the path to the illness is separate from the path back to health.
Yeah.
So just removing, for example, let's call it, let's say that a particular pesticide exposure was important during let's make it even a simpler thing. So we know that a gestational infection leading to high fever at the second trimester increases the risk of autism, gives you an odds ratio of about 1.53. So about 53% increased chance of that child developing autism by about age 3. That infection is no longer present, yet the child still has autism.
And by tapping down on the cell danger response, you can actually shift, potentially, in some cases at least, shift the change in mode.
Yeah, so it's really about changing the cellular message. So once the cell enters into the danger response, it is receiving a new set of marching orders, basically saying, and those marching orders, the world is a threatening place, you need to keep up your guard. And that's manifested actually even at a cellular level with changes in the biophysical properties of membranes and the properties of channels in the membrane that release and accept molecules. And so what we found, so I'll say this, and this will be not an easy concept, but I'll say that the original response to threat or to injury, is deterministic. The recovery from that injury is probabilistic. And what we've learned is that we can increase the probability that children can re-engage a more normal developmental trajectory by removing some of the signals that keep that cell danger response persistent. And one of the most fundamental ones that keep that signal persistent is extracellular ATP signaling.
Yeah. One final question, and we're running into another person's time, so if you can try to be relatively brief. This fascinating responses though. Is there data on the involvement of purinergic signaling genes in ASD? Is there data involved?
Yeah, actually so that there's, so we've, yes, so the short answer is, and there's a paper from 2012 that looked at brain RNAs in children with ASD that found that remarkably the genes that were involved in purinergic signaling were the most correlated with core symptoms of autism spectrum disorder.
Yeah,
so
fascinating.
Among many different genes that were analyzed. So yes, there are. That's work that's 8 years old, and there's a lot more now.